Chapter 10, Section 4 is on the structure of RNA and also on protein synthesis. RNA is a ribonucleic acid, and there are three types that we'll look at: mRNA, tRNA, and rRNA. The structure is also made up of repeating nucleotides, just like DNA is. Here's our nucleotide with our phosphate group, our sugar, and our base. But instead of being a double helix like DNA is, it will be single-stranded, and its sugar molecule will be ribose instead of deoxyribose. Finally, the bases are a little bit different. Instead of thiamine, there will be uracil. So A normally pairs with T. In RNA, you're going to have A pairing with U. So our four bases are adenine, guanine, uracil, and cytosine. And G and C will still bind to each other, and A and U will bond to each other. mRNA, or messenger RNA, is a single uncoiled chain, and it is going to carry genetic information from the DNA in the nucleus out to the ribosomes in the cytosol, which is in the cytoplasm. So when our DNA molecule opens up, we can use one side as a template strand to build this single strand of mRNA off of it, and then that will leave the nucleus and the DNA and go out into the cytosol to find a ribosome. Transfer RNA, or tRNA, is also a single chain, but it's coiled up to form a hairpin shape. I think it just looks like a T. So that's how I remember this is the transfer RNA. And in addition to these bases that you will also have an amino acid on the end of it, which is going to help to build our protein. So at one end is going to be our amino acid, and at the other end there's going to be a series of three bases in a row that's called the codon, and will pair with the anticodon. Ribosomal RNA is called rRNA, and this is actually the most abundant form. It's a single strand, but it's been all folded up so that it's in this big glob, and they actually make up the ribosomes, which is where the proteins are made. Transcription is the process of copying the genetic information from the DNA to the mRNA. So the mRNA is called a transcript. We're using that DNA template to make this mRNA strand. So this is occurring in the nucleus because that's where the DNA is located. There's also a process that I just kind of want to mention briefly here. It's called methylation, which is just the process of regulating gene expression, and this has the ability to stop this transcription process. So methylation is actually going to help in allowing the cells to specialize. The steps of transcription involve the molecule RNA polymerase binding to a promoter of a particular gene, and then that will begin the copying process of using the DNA to build this new complementary strand of RNA nucleotides. So using our base pairing rules of A with U and C with G, that mRNA strand will be made, and once that polymerase molecule reaches the termination signal, that will signal that transcription is done, the mRNA strand is complete, and it will be able to leave the nucleus and go out to the ribosome. The genetic code is the correlation between the nucleotides and the amino acids. So what bases you have and what amino acids they will code for. If you recall, there are 20 different amino acids that are used to make proteins or polypeptides. And the same genetic code is used by most organisms to translate their mRNA transcripts into actual proteins. This is a table that we're going to use to figure out what proteins are actually going to be made. The tables are set up so that the first base is going to be read on this side. It could be U, C, A, or G. The second base would be also U, C, A, or G, and then the third one, you also have the same four options of U, C, A, and G. So depending on what combination of bases that you have, say I have U, U, and U all in a row. That means that this is coding for phenylalanine. Okay, this is the amino acid. So that would be one of the amino acids that would be going to contribute to the protein that I'm trying to make. A codon refers to 3 mRNA bases in a row. Okay, that is called a codon. So they code for proteins. The start codon is the one that has to start this whole process. So it's actually 3 in a row of A, U, and G. So A, U, G in a row is a start codon, and that will signal the ribosome to start translating the mRNA molecule. The stop codon, there are actually 3 of, and it could be UAA, UAG, or UGA all in a row, and that would signal that the protein is finished and it's time to stop translating the mRNA, and the protein can be released wherever it's needed to go in the body. So this little animation down here, this is our mRNA strand. So 3 in a row is going to be our codon. Here comes our tRNA molecule. So AUG is start. So what's pairing up would be UAC. Here's our amino acid that's attached to the other side. Methionine starts it. This brown thing is our ribosome where we're located. So now we need the next codon would be red, would be GUA, and we would need another tRNA molecule to come in here and pair up, and this whole strand will continue until we reach a stop codon. So translation, which is also referred to as protein synthesis, is where we assemble the proteins from the encoded information in the mRNA. So the anticodon is located on the tRNA, and the mRNA has the codon. So they will be complements to one another. So here are the steps of translation. Our codon, which remember is our mRNA, is paired with our anticodon, which is on the tRNA, and the tRNA will then add the particular amino acid that we're coding for to the polypeptide chain or the protein. So if you're looking at this animation, here's our AUG from our mRNA. So that pairs up with UAC, which was our anticodon that brought methionine. We paired up our next codon with the anticodon on tRNA, and it brought valine. And the process is going to continue, forming these bonds between all of the amino acids until we finally reach a stop codon, at which point the protein will be released and sent off elsewhere in the cell. Now ribosomes, some of them are free floating in the cytosol, and some are attached to the endoplasmic reticulum, the RER. The ones that are free in the cytosol are the ones that are going to make proteins for that particular cell. The ones that are attached to the RER are going to be transporting those proteins out of the cell to other places that they might be needed. So there's a couple binding sites where the mRNA and the tRNA come in. So this smaller site is called the mRNA binding site, and then the tRNA comes into this larger portion up here, and that was where the tRNA anticodons will come and pair up. This is just a summary here of this protein assembly process that we just talked about. So the ribosome attaches to the start codon on the mRNA transcript, which is going to pair with the tRNA anticodon, and the ribosome will continue moving along that strand of mRNA and attaching those amino acids as the anticodons are paired up until the ribosome reaches the stop codon, and then the mRNA gets released and the protein gets released, and everything is finished.